Click for IB Biology. Here we're looking at the Hershey Chase Experiment of 1952. There's a blank PDF of this presentation down here in the description, along with a quick quiz, 10 questions with answers for you to check your understanding. And then at the end, there will be channel links to various other videos in the IB Biology sequence. So at the time of the experiment, remember that this is pre-1953 Watson and Crick. 1952, Hershey and Chase published their work. Both of these researchers had previously believed that it was proteins that carried the genetic information. They were skeptical about previous research work that had been done at this particular period of time, trying to determine what was the what was the genetic material. They chose to look at the reproduction of what is called a T2 bacteriophage. This is a type of virus, a virus that normally infects prokaryotic bacterial cells. So we got a bacterial cell drawn here for you. Now I've simplified the bacteriophage structure to show the outer protein structure called the capsid, and then in the center there is a nucleic acid. So at the time of the experiment, both Hershey and Chase believed that the genetic information would be carried on the apparently more complex proteins rather than the nucleic acid. And they were aware that in the biology of the bacteriophage, what happens is that the bacteriophage injects material into the bacterial cell, and then that material brings about the replication of the phage, which would require the production of proteins and of course of the nucleic acids. So their money was on the proteins. What they did was, prior to the phage actually carrying out its replication, in the protein structure they added a radioisotope of sulfur known as sulfur-35, and to the nucleic acids they added phosphorus-32. Both of these are radioisotopes that can be detected, and this particular technique that they used relies very much on the technology of having radioisotopes and of being able to detect them. And so the hypothesis was that whatever enters into the bacteria, that will be the material which actually brings about the replication. So the phage was allowed to infect the bacterial cell, and then in the next step, what Hershey and Chase did was they took the infected bacterial cells and they famously used a food processor to homogenize them, to smash them up into bits, and they put those homogenized pieces of bacterial cell into a centrifuge tube like so. And then the centrifuge tube is subject to very, very high speed spinning, and the effect of this is to separate out the different parts of the contents up here, which of course were smashed up bacterial cells. And what they discovered, or what occurred, was a pellet formed at the base, which was based on the bacterial structures, and up here in what's known as the supernatant was what was left, the lighter materials. And they discovered that what had accumulated in large quantities in the bacterial pellet was in fact the P32. It was necessary to use methods of isotope detection in order to determine this, but the bacterial structures actually contained the high levels of P32, showing that what was actually put into the bacterial cell was the nucleic acids. And so this added to the information, the suggestion, the evidence that nucleic acids were in fact the major, were not the major, but were the actual molecule of genetic information and not the proteins.